Hi, good afternoon, everyone. Thanks for coming. I'm excited about having today's speaker here today. So John Ioannidis, this is his second of 3 talks, so he's doing the Michigan Triple Crown here. So yesterday he talked to us a little bit about the role of bias. Today he's going to talk about precision health. And then tomorrow, as part of the Department of Internal Medicine Grand Rounds, he's going to be wrapping up that series as well, talking about reproducible and useful clinical research. So if you have the ability, try to catch all 3 of the talks. They're sure to be really remarkable. So I just wanted to start really just with a brief introduction. I mean, you could go on really like all afternoon talking about John. He's the C.F. Renborg Chair in the Disease Prevention Department at Stanford University. Where he's also a professor of medicine, a professor of biomedical data science, and a professor of statistics. And he also co-directs Metrics, which is their Institute on Meta Research, the study of how we should be doing studies. And John's had an interesting kind of career and path. He was born in New York City, but then grew up in Athens, and he's benefited from actually experiences on both sides of the Atlantic, but currently he's at Stanford University. And it goes really without saying that he's probably one of the most original and influential physician scientists of his generation. So I could go on and on with a specific report, but I'm going to share a particular story which I found kind of really compelling. So if you do know John, you know him through his magnum opus, right? His paper, Why Most Published Research Findings Are False, a paper published in 2005 in PLOS Medicine, and a paper that's been cited nearly 7,000 times at this point. So what's really fascinating about that story is when you ask John, like, well, how did you come up with that idea? How did you write that paper? It's a single authored paper. And so what I've learned over the last couple of days is he did it as most of us would do on a vacation in a Greek island. So he wrote that paper in Sikanos, which is a tiny island off the southeastern part of Greece. It's very close to other islands that some of you might know, Santorini, etc. But as opposed to those kinds of glamorous vacation destinations, this is an island that has about 250 people in it. And I just picture, John Ioannidis, like in 2005, over a period of 48 hours, typing out what has become, probably one of the most seminal works in kind of our scientific and meta research fields. So it's a real honor to have you here, John, and we look forward to hearing your talk.
Thank you. Thank you for the very kind invitation and the wonderful introduction. So I will try to share some thoughts on precision health, big data, evidence-based medicine. These are all terms that have very strong friends and very strong skeptics also probably among the audience and beyond. What is evidence-based medicine? We have to go back to the original quotes of the term by David Eddy in 1990. He said that consciously anchoring a policy not to current practices or the beliefs or experts, but to experimental evidence. And this means that the pertinent evidence must be identified, described, and analyzed. So you have experiment and some effort at synthesizing and putting together information and analyzing it. David Sackett probably has written the classic definition of evidence-based medicine. He says it's the conscientious, explicit, and judicious use of current best evidence in making decisions about the care of individual patients, and I underline the word individual, and that means integrating individual clinical expertise, again, individual is very prominent there, with the best available external clinical evidence from systematic research. So that definition immediately has 2 major components. One is an individualized approach, both at the patient level and at the clinician level, and therefore also at the level of their interaction, their encounter. And then a second component, which is science, information, evidence, the best possible evidence, the best possible science, put together in the most unbiased manner. Seeds of wisdom and of debate in these definitions. Experiment which practically means randomized controlled trial. Evidence-based medicine got almost synonymous with the advent of the need to get better and larger and more relevant clinical trials, randomized controlled trials. Systematic approach, systematic research, and that translated to systematic reviews and meta-analysis as being a tool for promoting integrated evidence. Individual patients, precisely so. So here's the roots that lead exactly to precision medicine and individualized medicine. And individual clinical expertise, again, extremely well aligned with what we're talking nowadays about individualized and precision health. Over the years, many hierarchies of evidence have been proposed, and the most popular ones have meta-analysis and systematic reviews at the top. Then you have randomized controlled trials following that, and very close to the level of no value or even negative value, you have experts, and even below that you have tweets or experts or powerful people tweeting all the time. Then you have multiple types of evidence that have evolved. Clearly, the traditional evidence-based medicine has been dealing with meta-analysis and randomized trials, so clinical types of information, but there's also observational evidence, there's mechanistic evidence, there's other evidence, and if you look across 180 million scholarly documents that are floating around with 20 million authors, there's lots of sand in that desert of information that a clinician is trying to go through on a daily basis, multiple times, back and forth, and a scientist is trying to make sense of and survive and get to some oasis of real discovery. We have also learned that evidence is less than optimal. Usually these pyramids are destroyed, like this poor destroyed pyramid in Abu Rawash. We lack the type of evidence that we want to have in place in order to have actions that we feel certain that they're going to do more good than harm. Also, pyramids can be bulldozed by property developers, like this one in Peru, and we have learned over the years that evidence can be severely affected by conflicts of interest of stakeholders who support, sponsor, and develop and disseminate evidence. Most of that is financial conflicts of interest, but increasingly we recognize that there's other conflicts of interest that could also be important in some settings. Some of them stemming from just genuine human curiosity, but still you can get a lot of strong allegiance bias even by people who otherwise have good intentions. There are alternative views to that standard picture of evidence-based medicine. And one of them that flourished in the mid-1990s was thinking that N of 1 trials should be at the top. Instead of having the composite picture from all the studies, all the trials that have been done, put together in a meta-analysis and a systematic review, we should just look at what happens at the single patient level. So N of 1 trials, Gordon Gwiat and others proposed pyramids that had them at the top, and many people favored that, but very soon we realized that N of 1 trials were not really doing what they were supposed to do. So N of 1 trials, which are currently proposed as the new wave of understanding and promoting precision health and precision medicine, actually are a design that was introduced in the 60s. We got its basic methods correct in the 1980s, flourished in the early 1990s, and abandoned about 20 years ago. And now they're being resurrected. Why were they abandoned to start with? Because they have these caveats. They're not good if the disease does not have a steady natural history. They're not good if there's carryover effect. They're not good if there are priming effects and if the effects depend on the sequence of previous choices that have happened or have been utilized in the same patient. They're not good if the disease has a fatal outcome in a relatively short course, because then you have no time to test multiple options and to choose which one is the best. And they're not good if there's poor or unpredictable compliance, adherence, tolerability, meaning if there's real life. Therefore, N of 1 trials met with all of these challenges and didn't really move very far. So here comes precision medicine or precision health, as I see that you prefer this term at Michigan, and we use exactly the same term at Stanford. I'm not sure which one is best. So what is that? I don't know. I'm supposed to be an expert in precision health, but I have no clue what it is, so I went to Wikipedia to find out. And here's the definition that Wikipedia gives. It's a medical model that proposes the customization of healthcare with medical decisions, practices, or products being tailored to the individual patient. So very prominently, again, the individual patient. We're back to the definitions of evidence-based medicine of the 90s. Now, the individual, if you simplify in some sort of semi-mathematical terms, is one of the population, is the other extreme of the population. By definition, precision medicine is thus aiming to have the most tiny and the most negligible impact possible at the population level. I mean, this is the starting point. We're trying to have an impact on individuals, therefore, we want to get the most negligible impact. Big data, again, I couldn't find a good definition, or I found probably about 100 different definitions on what they are, and this is my attempt to define big data. Big data is data that carries the least possible information content per unit. The more insignificant the content of information per unit, the bigger the big data. So why is that? Because if we really had information with a lot of content that is meaningful and useful, we wouldn't need big data. You know, why should we waste our hard drives and our time and our resources and our computational time if we could just measure one thing and that would be the answer to all our problems? The fact that we need to measure all these information units to try to build together something that is useful means that we are in a situation where we are struggling with situations with minimal information per unit, but hopefully so much information that if you multiply the per unit times the amount, hopefully it can still be useful. It's the exact opposite of Bradford Hill request of what should we believe. So Bradford Hill, one of the fathers of clinical epidemiologists, said that I'm willing to believe if something can be checked on the back of an envelope. If it's like 2 + 2 or maybe an odds ratio on a 2x2 table is fairly possible to fit in the back of an envelope, that's okay. If it's more than that, forget it. You know, it's too complex for an epidemiologist, let alone for an everyday clinical practitioner, let alone for a poor patient who tries to get some benefit. So how can we have these two opposites come together? I would argue that we can start from the premise that precision medicine or precision health is the study of the most insignificant. And then use one quote from one of my favorite poets, Odysseus Elitis, that you'll come to learn a great deal if you study the insignificant in depth. So precision health is a way to study the insignificant in depth. How deep can we go? Well, before we decide how much in depth we can go into the abyss, let's try to see what do we have in place and whether we can really have the bathymeter to reach the abysmal end of all these data. In 2018, evidence-based medicine has lots of data, not necessarily reliable, but we have lots of data both at the individual level and at the population level. Sometimes we have both ends of the spectrum being heavily armed in information. We have big and small, deep and shallow, broad and narrow types of databases. We also have the patient-clinician interaction that is still there, but is probably suffering much of the time because of limited time and because most physicians now have to deal with a computer and with data rather than find time to even talk with the patient. And we believe, and we have evidence, and pretty good evidence actually, that shared decision-making is a good thing. So if that information can communicate and can be shared meaningfully between physicians and patients, that would be really nice. What kind of information are we going to share with patients? Unfortunately, despite the fact that we have tons of information, very little of that is clearly useful. This is an analysis that we did a couple of years ago. We took 1,400 topics in medicine that had been assessed in the Cochrane database of systematic reviews, and of those, less than half, 43%, had grade summary of findings assessments. A grade is the grades of recommendation assessment, development, and evaluation tool that is trying to assess what is the quality of the evidence when you have data from one or multiple trials. What was happening in the other 57%? Well, mostly there was no evidence, and this is why there was no grade assessment. In these cases where we did have evidence, looking at the first primary outcome, only 13.5% of the time we had high quality of evidence. Even when looking at all outcomes that had been assessed and where the primary set of outcomes, only 19.1% had at least one outcome with high quality of evidence. If you limit your focus to the reviews that had high quality of evidence available and significant results, nominally significant, just with the typical 0.05 threshold, and a favorable interpretation of the intervention, meaning someone concluded, so this is a treatment that is good to use. Only 25 out of the almost 1,400 topics had this type of situation. So less than 2% of medical topics had high quality evidence, significant results, and someone said, yes, go ahead and do it. 98% of the time we had modest or very large uncertainty about how exactly to deal with populations, let alone patients. One confounding problem is that we don't have many discriminatory tools that can tell us which among these 98% of topics where we have uncertainty, we can lean more towards saying, okay, maybe we have some evidence to act, or maybe we don't have enough evidence and maybe we need more, or maybe we have some evidence not to do anything. The problem is that our typical tools that have been used for discrimination since the times of R.A. Fisher have become obsolete. Almost all scientific papers claim that they have found statistically and or conceptually significant results. Obviously, all of my grant proposals claim that what I'm planning to do will be highly significant one way or another, although I mostly submit mediocre ideas for funding. And if you look across the entire PubMed worth of abstracts and full-text papers, 96% of them report statistically significant results. This is an analysis that we published in JAMA about 2 years ago. We looked at close to 15 million abstracts in PubMed from 1990 until 2015, and close to 1 million full-text articles from PubMed Central. 96% of that literature claimed significant results practically whenever they had p-values of some sort listed. Practically all of them claimed to be novel. It looks as if discovery has become so commonplace that it's a boring nuisance at this point. So how can we tell what to use out of that huge mass of information? To make things worse, almost any result can be obtained unless we pre-specify what kind of analysis we're going to have, and the availability of big data is making that challenge even bigger. This is the Janus phenomenon after the Greek-Roman god who can see in both directions, and these are data from the National Household Survey. What I'm plotting for you here, for example, this panel, is whether alpha-tocopherol or vitamin E is associated with the risk of death. And there's a cloud of 1 million different results. This is the hazard ratio, and this is the minus log 10 p-value that are obtained for the very same question being addressed in the very same database. How do you get 1 million different options? Practically, death can be affected by many other variables. So for each variable that you can adjust or not adjust the regression, you have 2 options. If you try to adjust or not adjust for 19 options, 2 to the 19 is 1 million choices. And 70% of the results suggest that vitamin E decreases the risk of death. 30% of the results suggest that vitamin E increases the risk of death. So Janus is looking in both directions, and depending on what you enjoy the most, you can report that vitamin E is great or vitamin E is horrible. When results seem to be more credible, unfortunately, still we have the problem that most of those are not necessarily patient relevant. And one might argue that in an individualized precision setting, we need to ask patients one at a time. What exactly is relevant to them? So that would be ideal, and then we have like a different textbook of medicine for each patient one at a time. However, there are some outcomes that are important no matter what. So I think that death, for example, is a very important outcome regardless. Some people may still say, I want to die, please, get me to die. Possible, but I think that still, even for that patient, if we can make him change his mind and let him live longer and live a good life, that would not be really that bad. So are there some outcomes that would be ubiquitously important so that regardless of the particular patient, they would be interesting to know what we can do with different interventions at the population or individual level? There's initiatives like COMET that are trying to put together such a list of outcomes that seem to be essential to study for different diseases. And in the case of preterm infants, where for good or bad the patients cannot even express their wishes on what is the important outcomes for them, things like chronic lung disease are clearly very important. You can measure many, many other things, but chronic lung disease is a major problem for preterm infants. So you want to know what different interventions would do in that regard. When we checked more than 1,000 trials on preterm infants, less than a third of them actually reported on chronic lung disease. 2/3 plus did not mention something that was so ubiquitously important to know about. Another premise about precision is that since we're talking about one patient at a time, hopefully the effects associated with that intervention should be really large. If these individualized interventions have tiny effects, who cares? We've had so many tiny effects floating around. The big promise is that now we'll put together biology, lots of data, informatics, complex analysis, black boxes, and we'll get you a huge benefit that if you have that profile, you will really do much, much better. What does our previous experience telling us when we look at evidence across medicine in terms of how often do we see large effects? This is an empirical evaluation we published about 5 years ago in JAMA, where we took every single meta-analysis that we could track in the Cochrane database. There were 85,000 meta-analyses that we could track, and we asked how often do we see treatment effects that are large and that are also replicable, meaning they're seen in more than 2 studies, and hopefully they have good statistical support, and hopefully they also have no clear bias that is visible and that would invalidate our trust in them. What we found is that large effects are very common, but they're very common in very small early trials, and very few of them survive scrutiny. So if you ask for a mortality effect where you have a 5-fold reduction of mortality risk that has been seen in 2 trials with a p-value of less than 0.001 and with no florid evidence of something being wrong in the evidence, along these 85,000 meta-analyses, there was only one topic, extracorporeal oxygenation, that achieved that kind of gold standard status of huge benefit clearly. Now, is that all? Are there no other interventions that are as effective as wearing a parachute when you jump for an airplane? Yes, there are more in medicine. Obviously, if you have someone with diabetic ketoacidosis and you don't give insulin, it's like letting him fall from the airplane without a parachute, or, well, depending on how serious it is, maybe jumping from the 10th floor, but a small chance of survival. There are such, but Paul Klaziou has created a list of such huge effects that have been thoroughly validated, large benefits, clearly so, and the list includes about 20 interventions, maybe 25 at the most. What is happening is that we do see lots of large effects, like odds ratios of 5+, even for things of mortality sometimes, but they're seen in very small studies, on average with 12 participants worth of data. And whenever we perform yet another study, the effect goes away. It either completely evaporates or it becomes much, much smaller, so then it's questionable whether it's really worth it or not. So based on a lot of experience the data that we compiled here included about 250,000 of randomized trials. We know that very large effects are not uncommon to find when we're dealing with small numbers, which is the typical recipe for most of the studies that are currently being done in precision medicine circles. But most of those really need to be validated before we can be certain that they're not flukes and would not disappear. Quality could also confound the picture. Quality problems are prevalent in clinical research, much like in other disciplines. Some of them may be unavoidable. Sometimes masking, for example, is not an option. Randomization very often is not as proper as it should. Allocation concealment sometimes cannot be guaranteed, and when it can be guaranteed, it has not been adhered. We have lots of evidence, and this is summarizing the result of the Brando project where I was involved, where we performed a meta-analysis, including several thousands of clinical trials and several thousands of meta-analysis. Practically, we concluded that if you have problems with randomization, if you have problems with allocation concealment, if you have problems with blinding, you're likely to get, on average, inflated effects. However, the average distortion is relatively small compared to the heterogeneity in the distortion that you can get. Most of these distortion effects are much larger compared to the precision effects that we're chasing currently. So unless we can fix these quality problems, we don't really know whether these effects that will emerge will be true or would be spurious. And even worse, because of the large heterogeneity in the amount of distortion that these biases introduce, we will not be able to just correct by saying that, well, we failed 3 aspects, we didn't have randomization, so let's correct by dialing the effect size by 10% and we will be fine. It will be 10% on average, but it could be 80% in some case. It could be even in the opposite direction in some other case, and this is impossible to really know. Another confounding factor. Much clinical research nowadays is done outside the US and Europe, meaning countries that have a tradition of research, in particular of clinical research. A lot of research is done in China, a lot of research is done in Eastern Europe. Very often the price for running these studies there is very low. You can run a clinical trial with 1/10 or 1/50 sometimes of the cost that you would need to do it in Michigan or at Stanford, where the cost would be prohibitive. Are these results unbiased? Here's another empirical evaluation where we looked again across all the Cochrane database in situations where mortality outcomes had been assessed in European and American trials, and also in trials done in countries that don't necessarily have such strong tradition of clinical research. Here's one example. If you look at calcium antagonists in aneurysmal subarachnoid hemorrhage, European and American studies show practically no significant benefit. It's very small if there's anything there, and clearly nowhere close to even a nominal level of significance. If you look at a small study in China, there's an 85% reduction in the odds, suggesting that this is a tremendously effective intervention. Is it that this study is completely flawed because it was done in China? I think not. If you look carefully, its quality scoring may look pretty good. But probably what's happening is that there's many, many more studies that have been done in China or in Eastern Europe or in other countries, and then they're trying to get through the bottleneck of getting published. And if you get a study that has completely negative results in the US, you know that it's not so easy to get it published in the New England Journal of Medicine. Well, probably you can get it published in some specialty journal. If you get a negative result in China, you will not be able to publish it anything, number one. Number two, you will not get a financial bonus. There's bonuses given by the Chinese government and institutions that could be up to $100,000 for a paper in Nature and a few $100 for something that you can publish in a more modest venue. And three, you run the risk of being flogged with a whip because you found a negative result. So you have to take into account what is the research environment in different communities and how that might shape the dissemination of results from small studies that may seem to have extravagant results. On average, studies from less developed countries had an inflation of the odds ratio for mortality of about 15%, which is larger than the average treatment effect of the most effective treatments that we have to curtail the burden of death. Here's another example from the Top Cut study where investigators, and this from the New England Journal of Medicine, realized that spironolactone behaved very differently in American and European sites versus sites in Russia and Georgia. And trying to go into more depth, they realized that the experience of the patients in Russia and Georgia was entirely different. Apparently, probably these people had not even been treated with spironolactone or whatever was the assignment they were supposed to be. Further confounding by sponsor bias. There are some types of clinical trials that almost always favor the sponsor. A couple of years ago, we looked at 57 non-inferiority trials with head-to-head comparisons. We took the largest ones, and we found that 55 of them showed results that were favorable for the sponsor. The success rate was 96.5%. I would argue if an experimental design has a success rate of 96.5%, is that anywhere close to equipoise? Why do we need these studies? We can get rid of them. We can say, well, the study will be successful, the drug will look very nice, and move to the next step. What is wrong here? Is it that industry-sponsored trials, and this is becoming more and more prevalent in the case of precision interventions, are biased? Is it that the industry has evil people who are cooking up the data and disseminating false results? No, if you look at the quality of these trials based on our traditional checklist, they look very nice. The problem is that the design is such that it's trying to optimize the chances of getting a nice looking result. So choice of comparators, choice of the non-inferiority margin, choice of the setting, choice of the exclusion criteria is such that you're almost doomed to get a nice looking result. And this is very difficult to decipher. It's a whole science behind the science of how to always get nice looking results. Further problem, can we trust the data? This is 329, and it's not a submarine, although it sounds like a U329. It's a randomized trial that resurfaced after 15 years after its submersion. So when it had first appeared, it was the pivotal trial that suggested that in major depression in adolescents, paroxetine and imipramine, two antidepressants, were very effective and very safe. 15 years later, independent investigators got hold of the individual level raw data, and they reanalyzed the trial from scratch, and they concluded that both paroxetine and imipramine are not effective and are not safe. So entirely the opposite conclusion. Can we trust the data? We want to trust the data. Unless we can trust what we read, we are really into deep trouble, because then how can we tell which cases are like 329 and which cases we can trust? Moving forward, here's a larger evaluation of reanalysis of raw data in individual trials. We tried to unearth every single case where a paper had been published reanalyzing an original trial for the very same clinical question, but it had been done in a separate publication than the original. We found 37 such cases in that paper that we published in JAMA in 2014. 35% of the time, the conclusions of the reanalysis were entirely opposite or very different compared to the conclusions of the original analysis. The original had claimed that the drug is effective, the reanalysis that it is not effective, or vice versa. The original had claimed that this is the subgroup, the characteristics of patients who need to be treated. The reanalysis said, no, this is the characteristics of the patients who would benefit from treatment. What is going on here? Is it that we have rogue reanalysts who are trying to make a fame around their name and are trying to put the original investigators into disgrace just by manipulating lots of fancy reevaluation of the analytical space? Actually, these papers are almost always published by the same original investigators who published the first paper. But it is happening in an environment where if you spend extra time to reanalyze your own data, you cannot publish a second paper. You cannot say that I spent extra time and had a second look and I get the same result. Can I get a second paper? Conversely, if you can get a different result, or if you can say that you get a different result, then you can get a second paper. It sounds very confusing, but this is the incentive structure that facilitates this feeling of irreproducibility in that setting. Here's a very different environment where raw data are available routinely. Two journals, PLOS Medicine and BMJ, have routinely made availability of raw data a sine qua non, a prerequisite to publication, and they encourage that all data need to be shared for any trial that they have published in the last several years. So we communicated with the authors of all these clinical trials, and we asked them to send us all the raw data from all the trials that they had published in these two journals. We got 46% of them. Oh, is that half full? Is it half empty? It's close to half, though. And then we spent time to reanalyze all these clinical trials from scratch to see what results we got. That was not bad. It's a pretty complex graph here, but all these points are very close to the diagonal. If they were exactly the diagonal, it would mean that we got exactly the same results. There were a few errors here and there that we got. I mean, we got in touch with investigators. They said, yeah, thank you for picking this up. But none of these cases was such that the conclusion of the trial would be invalidated. So in a different environment, in a different culture, where everything is supposed to be shared, where you have journals that have very strong enforced policies, where you have the most transparent investigators who want to do that and go through that policy, and you have even the subset of those who are saying, here, go, take my data and reanalyze them, almost everything looks fine. So is it that the culture is what matters, or is it that we just deal with different types of selection biases and selection processes? Another promise is that since we have all these problems with the traditional study designs that depend on availability of randomization and huge expenses, maybe we can replace them with routinely collected data. So there's tons of routinely collected data-based analysis that try to estimate treatment effects, and our methods are becoming more and more sophisticated to try to incorporate patient characteristics that define populations that can be almost matched to a randomized equivalent. So these are results of routinely collected data studies using propensity matching or propensity score adjustments versus randomized trials that were done afterwards. So when the RCD study was released and published, there was no randomized controlled trial, so that one could compare notes, but then subsequently there were randomized trials published, and these are mortality outcomes. The average difference is a 31% difference in the odds. It's huge. It's like 3 times the average effect for mortality that we see even for very effective interventions. So if really this is that bad, clearly that's not a panacea for replacing the paradigm of randomized trials. Another solution is to build prediction tools, prediction models, prediction algorithms into mapping the individuals or the subgroups or the substrata that would do better or worse or have different magnitudes of treatment effect. And I'm very fond of that. I'm doing quite a lot of research in that space. I still hope that something major will arise, but I think most of my papers are probably not much worth. This is looking at the field of cardiovascular disease risk prediction, and this is the number of articles that present new predictive models for cardiovascular disease. There's about 400 different models that have been presented in the literature for cardiovascular disease. And as you well know, when ACC, AHA decided to release new guidelines, they looked at available predictive models and they said none of them is good enough, so they developed yet a new one that everybody feels that is one of the worst models. You know, it's not calibrated, it's completely misaligned with risk levels and so forth. So how can we really find predictive tools that would be validated but also useful, and that there would be some consensus that people would like to use? The alternative approach is that why should we have everybody using the same model? Maybe each hospital, each center can use their own model. So we have a new wave of lots of studies that take electronic health records and they develop a local model that actually can be reevaluated and updated and upgraded and changed in real time as we get more information and more data. So this is an empirical evaluation that we did with Ben Goldstein and his colleagues at Duke. We looked at EHR-based predictive models, and this is the sample size that they used. As you see, some of them go up to 1 million plus of individuals, which is very good. I mean, the problems of small studies that we were facing with randomized trials clearly is gone. We are facing the exact opposite of just having overpowered situations. The number of events, again, very nice. This is 10,000, sometimes even more than that. And the number of variables, sometimes we can include 1,000 variables, even 10,000 variables in the model. Try to visualize and to communicate to a physician or to a patient. This is a model with 10,000 variables that we have included. That would take probably 4 years to explain. So you just say, it's really good. Trust me, it's going to be fine. How well do these models do? They don't really do that well. I have to say that I was disappointed. I was expecting to see some AUCs or some reclassification rates that would be much better than what we see. On average, the AUCs were like 0.7, like the oldest models that we had in the 1940s when we could only measure CBC and creatinine. So having lots of variables, I'm not saying it's a bad thing, but most of these models don't really add up much in terms of predictive ability. I don't need to remind you that subgroup analysis have a tradition of leading us down the wrong path. There's a lot of discussion for many years, even preceding precision medicine, about using subgroup and effect modification and stratified medicine. And the classic example probably is from that paper in the Lancet where these are the months of birth. So this is the zodiac cycle horoscope, and this is the absolute risk reduction from carotid endarterectomy in people with symptomatic stenosis. Clearly huge subgroup differences, heterogeneity p less than 0.0001 based on the zodiac sign, but obviously this is 0% likely to be true. If you look across the literature of such subgroup claims, which are like the first step to inform some sort of stratification towards personalization, most of the claims that have been made in the literature cannot be reproduced. We published a couple of papers where we looked at all the sex-based subgroup differences across all Cochrane meta-analysis that we could identify in randomized controlled trials, and we found very little that was reproducible across multiple trials. There were lots of sex differences. If you looked at single trials, they were just very common. But something that was seen again and again, very uncommon. How about if we combine lots of databases, like lots of randomized trials? If we perform meta-analysis of raw data, of individual level data, and then try to identify closely validated subgroup differences. So this is another empirical analysis where we looked at all the meta-analysis of individual level data that have been done to date, and these are the results for subgrouping variables that seem to discriminate between different groups that have different behavior to treatment. These are the p-values for individual level subgrouping variables and for group level where the entire group has the same value, for example, the type of dose that is being assigned. P-values of 0.05 or close to that and 0.01 or close to that, really not very nice looking. If you translate them to base factors, they translate to base factors of something like 2.5 to 5. For a Bayesian perspective, this is okay to mention, but is it likely to be true? No, not really. I mean, probably a small minority of those may be true. If you look at the magnitude of the effect modification, so how much difference in the treatment effect do you get in people who have different types of covariates, the average magnitude is less than 0.2. 0.2 traditionally in a standardized mean difference scale is the threshold for a small effect. So the effect modification on average is less than small. Eventually, we want to get something that's useful. This is what evidence-based medicine was supposed to do. This is what precision medicine or precision health is supposed to do. But getting useful clinical research is not easy. In that paper in PLOS Medicine 2 years ago, I tried to come up with 8 criteria or features of what we really want. And it's the same regardless of whether you believe in evidence-based medicine or in precision medicine. First, we need to have a problem. We need to have a problem to fix. If there's no problem to fix, just creating problems, creating diseases that don't exist, just moving the definition so that everybody becomes sick and needs treatment, that's not a good idea. Second, we need context placement. We need to know what we already have available in terms of information. Maybe we have none, or maybe we have 522 trials, as we do in antidepressants. And then, why do we need another one? We need information gain. Is that new study, small, big, randomized, non-randomized, going to tell us something? If not, or if it's going to tell us something only if it gets a particular type of result, this is not a good way to decide to do it. Pragmatism, does it reflect real life? Or if it deviates, does it matter? Patient-centeredness, does it reflect our patient priorities? Have we asked patients what do they really care about with the background disease that they have? Value for money, is their research worth the money? There's formal ways to assess that with proper tools, but it's very rarely done. Feasibility, can it be done? About 35% of randomized trials in surgery are abandoned because of futility. They're assuming that they can get 50 patients on board, but after 6 months or 1 year, they have only enrolled 4 or 5. And finally, transparency. Are the methods, the data, the analysis verifiable and unbiased? And this is where reproducible research, open science comes into play. Where do you find these studies? If you look across the literature, most papers that you will see circulating in respectable journals don't meet more than a couple of these criteria, or even none. If you decide just to read New England Journal of Medicine, Lancet, JAMA, like the top of the top, again, most papers will meet very few of these criteria. And even though they will be better on average in fulfilling some of these requirements, most of the good studies would not necessarily be there. Do we need to speed up or do we need to slow down? That's another question. Facing these challenges, one option is to say, well, we have all these problems. Thank you for mentioning them again. You're such a bad guy to mention them. But the way to move forward is to just get as many interventions out there, get them licensed, and we'll sort it out. We'll let the dust settle. We'll do some studies after the fact, and then the real winners will emerge. This is actually an idea that is not new. It has been going on for over 10 years now. It's very prevalent in cancer, prevalent in HIV. I think HIV is a nice success story. I was at NIH when we run the pivotal clinical trial, ACTG-320, that showed that you can have a treatment that can completely change the course of disease. Huge success. And some other diseases have this kind of accelerated approach. What have we learned? Well, first we have learned that the studies that are supposed to be done after the fact, they're not done. Once you have something licensed, then it will go mostly into the mode of being evaluated for yet new indications beyond the ones that it has been already approved. Nobody will go back, or very few people will go back and try to make sure that what we approved it for is really valid. These trials of using that new treatment as background, meaning that they take it for granted that it works, but then they build additional new interventions that are also eager to get licensed, are happening very fast. Within 1 year, we're shifting to yet the new target being licensed. And if you compare the time of the trials where the intervention is tested for the initially approved indication versus other indications, there's hardly any difference. People take it for granted that since it got licensed for something, it's good for everything, which is exactly the opposite compared to the acceleration happening with a precision mentality in mind, that we're proposing accelerated approval because it can really affect that particular type of individual with these specific characteristics. The other mode that is becoming more common is to approve based on non-randomized trial data at all. And this is an analysis that we did with all the European Medicines Agency approvals over the last 10 years, and we're doing the same with all the FDA approvals. Roughly 10% of approvals of new medications and biologics have absolutely no randomized data. If you look at the effect sizes, some of them look pretty big. So this is absolute risk difference, and this is odds ratios. And on average, you see some fairly large odds ratios. So something like a 12 odds ratio is really nice, but the effect sizes could be all over the place. Sometimes they're close to 0. There's still drugs that get approved with non-randomized data with effect sizes that suggest absolutely no benefit, but there's some biologic rationale that people defended with that, well, it works on that mutation, and we've seen that this to be important, so therefore it must be effective. Systematic reviews and meta-analysis are not going to help necessarily much in that situation. We have lots of them. We have exceeded 100,000 published meta-analysis nowadays. It's more of an epidemic that is evolving. But I think that if you have this type of starting block of information, they're unlikely to be useful. We start seeing some weird phenomena. We see, for example, China becoming a champion in meta-analysis because, again, you can get some money out of each paper that you publish there. And this is genetics, genetic meta-analysis. Nothing was being published from China, but in 2014, they represented about 70% of the global production in English language journals, in serious journals, and now they're about 85% of the global production. There's lots of contractors that publish meta-analysis, and tons of them. There's 100 companies that you can pay and get a meta-analysis, and then if you're the industry, you can decide to publish it or not, depending on what the results look like, and it will be very nice looking. You pay a little bit, but that's not a lot of money compared to other areas of research and development. As a result, we get tons of meta-analysis, like these are network meta-analysis, which is the most difficult design to do, but in some cases we have up to 28 meta-analysis on the same topic, like biologics for rheumatoid arthritis. And none of them is exactly the same. They cover different treatments, and none of them gives the same coverage. None of them covers more than 50% of the studies that have been published, and none of them agrees with each other. These are the results. Again, if you want to pay, you can get a meta-analysis to give you the result that would fit your agenda. This is one practical example, since I'm surely got you depressed with what I have been describing, which is the best antidepressant to use. These are 4 network meta-analyses done by very good friends of mine. They're the best meta-analysis in the world. I know that because they're my friends. So I want to boost them before I destroy their work. Paroxetine, according to this meta-analysis, is the best antidepressant. According to that one, is one of the worst. Sertraline is the second best here, is the worst or one of the worst here. Choice of how exactly it's going to be done can make a difference. In a way, meta-analysis can be the last step of a marketing tool that you can get the conclusions that you want to see. Among 185 meta-analysis of antidepressants for depression, when an author who was an employee of the manufacturer was involved, there was 22 times higher chance of not having any negative statements. Actually, there was only one case where you had industry involvement and some negative statement, and I'm not even sure that these people are still employed by that company. So only 3% of meta-analysis are both decent and clinically useful. I think this is the best design, and when it works, it can help a lot. So evidence-based medicine is not dead. When you identify these 3,000 meta-analysis papers, they can really be helpful, and they can tell you what to do for population averages and perhaps also for a little bit of individual effects, but they're not that common. Last possibility, maybe getting from the individual effects much larger population effects for the same pathway. The classic example here is PCSK9 inhibitors, where seemingly we have a very nice success story. We started from a genetics discovery. We identified a gene that seemed to be very important in familial hypercholesterolemia, and it also had variants that would affect the risk at a population level. Then developed a drug that was based on that target, and we see that, wow, we can really bring LDL down across the population. Was that really a success story? If you look at the price of that intervention is so high that clearly its cost effectiveness is not something that is desirable. And obviously, we still don't know whether the benefits in lowering LDL and in some clinical outcomes would also translate to mortality benefits, but apparently doesn't seem to be so prominent. So I would close by saying that we may need to reverse the paradigm, maybe move from poor primary data, retrospective reviews, and this type of fragmented information to prospective meta-analysis as the key type of primary research. Think about what are the problems that we want to solve and design a clinical agenda where everybody working on that field, we're joint forces worldwide, the data will be incorporated prospectively. They will be individual data, but they will contribute towards the same overall analysis. We can design what type of comparisons we want to test. We will design the next study based on what we have in the composite evidence until now. I think it's unlikely we will be able to fuel precision medicine or health on individuals until we can obtain large-scale coordinated evidence on large populations to start with. To conclude, evidence-based medicine has been with us for a long time. It has acquired tremendous power, but most of the medical evidence is either problematic, spurious, or false, or has no utility for medical and shared decision making. The main utility of systematic reviews and meta-analysis has been to reveal problems with the biomedical evidence. Precision medicine and health aims to satisfy one of the main pillars of EBM, to deal with individuals, which is nice, but by definition is likely to have minimal impact on life expectancy and other major population outcomes. Still, a synergy between large-scale evidence and precision approaches would be useful to tell us what we can learn from both. Expectations of replacing experimental randomized evidence with non-randomized data need to be tempered. I'm not saying that it cannot happen, but most of the time we will need randomized trials. Conflicts of interpretation need to be minimized for primary data, for trials, for meta-analysis, and for guidelines. And perspective building of a research agenda may become the gold standard, hopefully, for primary clinical research, both at the individual and the population level. Special thanks to some of my colleagues who contributed to some of the work that I shared with you today, and special thanks to all of you for tolerating me at 5 p.m. Thank you.
Any questions? I can start with one. So John, you've been thinking about these issues for several years now, and yeah, painting a little bit of a depressing picture. Do you think that in some ways, like this movement towards precision health and big data will actually solve some of these issues, just kind of in a general sense, or do you think it's going to be fuel that's just going to add to the fire and it's going to make these problems even like many-fold worse as we think about things over the next decade?
I think it's up to us. It can go either way. I think that if we realize the challenges and build on circumventing them and generating more openness, more transparency, reduced conflicts, larger databases with more accurate measurements, we have a chance of addressing some of the long-standing problems that we have not solved with clinical research until now. Conversely, there's a great opportunity of having more data and therefore more errors, rather than more discoveries and more useful observations. So I don't want to have someone come out of this as we're doomed and nothing can be done. We have lots of opportunities, we have lots of tools, we have lots of possibilities. The question is how exactly are we going to use them to get something useful? So these 8 criteria could be some sort of guidance of how do we use these new tools.
Thanks. Actually, I'd like to build on Brahmaji's question a little bit. And this actually comes more from your talk yesterday, but it relates to this talk as well. I work in implementation science, implementation research. And at the end of your talk yesterday, you outlined a number of steps that could be taken. Things could be implemented. And in a sense, your criteria are also things that could be implemented. But I'd like your thoughts on what it will take to do that. If you think of implementation as behavior change, and you think about the scientific and medical research complex in this country and internationally as the substrate of people whose behavior needs to change, what will it take to actually get that change to happen?
So I think to achieve change, you need to have the major stakeholders aligned. And the major stakeholders are scientists, funders, journals, professional societies, institutions, and also the general public and patients could be quite influential. You don't necessarily need to have all of them aligned to do exactly the same thing, but they need to be sensitized and need to recognize that these are issues that need to be tackled, and it's important to move in that direction. Once you have some of these stakeholders moving and the others will follow, if you have just one, it's not going to happen. So no single journal alone can change the world. No single scientist alone can change the world. But if you have multiple stakeholders, you will see things happening. And every successful transformation, like registration for clinical trials, happened when you have multiple stakeholders being aligned. So you had both journals saying that I'm not going to publish your paper unless it is, if it's a trial, unless it's pre-registered. And you had funders who would say that you need to do that. And you had Clinical Trials Gov saying that here is how to do it, and you have to do it. And you have regulations trying to incentivize and ask for it. So we need, I think, a critical mass for these changes to happen. Data sharing. it was very rare, and this is why we only came up with 37 reanalyses that had been published in 2014. Right now, there's more than 10,000 clinical trials worth of data that someone could access as raw data. It's still a small portion compared to roughly 1 million, but I think we've seen some action. Statistical tools analysis, again, we see some evolution over time. So I'm not necessarily pessimistic. I think we do see changes, we do see some paradigm shifts. At the same time, our challenges are becoming incrementally and sometimes geometrically more pressing, because we have more data, more analysis, more people eager to create these sort of results.
Can you comment a little bit about how heterogeneity can influence the results and how it can be incorporated into, let's say, meta-analysis or trial and error? One of the stories I found mind-boggling is for about 20 years, there was a drug that worked in a very specific kind of lung cancer, and imitabine, I forgot the exact name. It was acting on the tyrosine kinase domain of EGF receptor, and it worked really well. In about 3% or 5%, or depending on ethnicity, etc., of cancer patients, it failed every single clinical trial, and therefore for a long time, in Europe it was allowed to prescribe in certain types of cancer, but in America it was only allowed to be taken by patients who had positive reaction to it before. I believe that has now changed. My worry is that with your example of, antidepressants or whatever, that there may be tons of such drugs that work really well in 3 or 5% of patients, but that are failing clinical trials, and the only reason this particular one was used is because we understood the mechanisms and it made sense. Mm-hmm. As long as we don't understand the mechanism, there may be such heterogeneity all over, and we may just be throwing out all those drugs because they only work in a subset.
So I take your point, and I think that it's likely that there are drugs that heterogeneity is masking effectiveness for specific subpopulations that we don't know the mechanistic substratum that would be the answer to that. I don't know how common that is, though. For example, cancer is the one discipline that has probably made most progress today in terms of applications of these sort of personalized treatments that are based on this type of biological heterogeneity, where you have a mutation well matched to the treatment. But you can see that in the super umbrella trial, the NCI match, only about 2.5% of patients with cancer can be matched to such a mutation that would be recognizable, and you can have molecularly targeted treatment. So even in a pretty successful paradigm, it's still a small minority. If you look at across all cancer trials at the moment in oncology, there is about 150 trials that have personalized designs in process, not finished, but registered in clinicaltrials.gov. So basket trials and umbrella trials and personalized designs that might fit to that concept, as opposed to about 50,000 trials that just have the typical bread and butter average treatment effect approach. We need to do more of those. So we need to go after these type of matching biology, matching mechanism to understand heterogeneity. But I don't want to also reach the other conclusion that in every case we will be able to find something. Antidepressants, for example, have been out there for more than 50 years. We recently published a meta-analysis in Lancet with 522 trials and 120,000 people. If you look at the literature, there's hardly any biological marker that you can reliably use to individualize treatment. I mean, it's like a holy grail. We want to individualize, and there are some possibilities, but nothing really that is as rigorous as a mutation linked to a specific biologic monoclonal antibody in cancer. So I think the challenges are different. Maybe some cases we're just looking down the wrong path. Maybe our whole thinking about what type of treatments we want to go after is wrong. In other cases, maybe we are in more solid ground, and we need to be open to challenge that.
One more question? Anyone? Right over there.
John, have you found any sets of trials where people have taken advantage, especially with cancer trials, of the evidence of differential expression of splice isoforms in key steps and key pathways? You know, there's this notion that by transcript analysis, sometimes called gene analysis, which it's not, that you can recognize that one of the biggest evolutionary developments is the exon-intron structure of genes and the splicing that goes on to produce multiple products from each gene, and then you just lump them all together and assume that they're all equivalent, which they are generally not. So I believe a good bit of the variation in trying to confirm biomarker result, which is generally fruitless, or to reliably treat even for a mechanism where there's a lot of variation in response, must lie with this splicing variation that is neglected.
I think it's a clear possibility, and it's a mechanism that I think has not been explored to its full potential. I would argue it still needs to be matched to problems that remain unsolved. You know, coming back to the very first criterion or feature of what we really want to get, because in terms of tumor biomarkers, there's about 2,000 new tumor biomarkers proposed every year. And we had actually published a paper a number of years ago where we found that 99% of that literature, so even higher than the average 96%, claim significant results. And even the few that don't claim significant results, I remember that 1% included a paper that had 125 p-values of that biomarker scattered throughout the text and tables, none of them close to even 0.05. But the conclusion of the abstract was that this is a very important tumor biomarker, as we have shown in our previous study. So we have lots of leads, and some of them are more exciting than others. But we don't have a very rational way to prioritize leads that would be more fruitful. And we just drown in a sea of tens of thousands of biomarkers, only a couple of dozen already adopted and used in clinic, and a very big space of a gray zone that is unexplored and very fragmented. So I think that this is one such example where you may have a clear winner, but it's just lost within that space of noise.
That's wonderful. So maybe one more